Great, thank you very much. I'd like to thank the organizers for the opportunity to present this work, and obviously the Mayo Metabolomic Resource Corps for the opportunity to do this work. So our lab is particularly interested in insulin action and insulin resistance and how these contribute to diabetes and other metabolic diseases. And the project essentially started from observations in the literature, some of which have already been mentioned in this the series of talks before me, that circulating branched-chain amino acids are altered in insulin-resistant states. And as such, previous and more recent studies have shown that in humans, branched-chain amino acids can predict the development of diabetes years prior to disease onset. Skeletal muscle is the primary depot for the organism for amino acid storage in the form of protein, which can be released upon stress states, both illness and starvation. And we know that insulin and IGF can affect not only glucose metabolism, but also protein synthesis and degradation in muscle. And lastly, skeletal muscle insulin resistance is a cardinal feature of type 2 diabetes, and this also can be detected up to 10 years before disease onset, or in folks that are at risk for developing diabetes, either through family history or other means. And so these generated our central hypothesis that perhaps muscle, insulin, or IGF resistance could lead to an imbalance of tissue and circulating amino acids and other metabolites, which may confer increased risk of metabolic disease. So here I'm introducing the canonical insulin and IGF signaling pathways. Actually, both ligands interact with highly homologous tyrosine kinase receptors, and they end up activating the same intracellular signaling cascades. The two primary ones I'm showing here are the PI3 kinase AKT pathway and the MAP kinase ERK pathway. These affect multiple downstream cellular actions, including protein synthesis and growth, glucose uptake and metabolism, suppression of atrophy and autophagy, specifically in muscle, and also proliferation in other tissues. And to try and get a sense as to whether these changes observed in humans could be coming specifically from the insulin resistance in muscle, we used a mouse model where we generated mice that specifically lack either insulin receptors or IGF receptors in muscle using Crelox technology. In here I'm just showing the mouse models that we used. We bred a skeletal muscle-specific ACTA1 Cre by insulin IGF receptor floxed mice, and we get out control mice which lack the Cre, either insulin or IGF single knockout mice, again knocked out only in muscle, or the muscle-specific IGF insulin receptor knockout mice, which I'll either call the McGurko or double knockout mice. It's important to note we've done a lot of diabetes and glucose metabolism in these mice, and we can show that each of these individual knockout mice do not develop glucose intolerance or diabetes at a whole body level. They are insulin resistant at the level of the muscle, but perfectly sensitive in the liver, fat, and other tissues. Lastly, to compare some of the metabolite profiles to a more classic model of insulin resistance, we fed some littermate control mice either the standard chow diet, which all of them were on, or a high-fat diet. So what is different about the individual insulin and IGF receptor knockout mice? It turns out that muscle size and muscle growth is the chief thing that has changed. What I'm showing on the left is the comparison of body weights between control mice in white, insulin receptor only knockout mice in green, IGF knockout in pale blue, and the double knockouts in red. And you can see that there's a 40% decrease in total body mass. This is primarily accounted for by a loss of muscle mass, shown on the right-hand side, with dissected weights of tibialis anterior muscle. Again, the most striking difference is that when you knock out both receptors, you get a 60% loss of muscle mass, and this occurs in all muscles that we've dissected from these mice. So the balance between muscle growth and muscle atrophy occurs via a balance between protein synthesis and protein degradation by either the proteasome or autophagy lysosome system. Our investigations into this McGurko double knockout mice, which shows markedly atrophied muscles, has shown that protein synthesis is actually increased, but protein degradation is much more highly increased. And this occurs both by the proteasome and autophagy lysosome system. In this particular study, we were able to use an inhibitor of the autophagy lysosome system to see this contribution to the branched chain amino acid pools by treating the mice with colchicine for 2 days. And again, this specifically affects the autophagy lysosome system. So what I'm showing here are the muscle tissue branched-chain amino acid metabolites. And to simplify things, I'm just showing the saline-treated controls first. We can see that control mice, compared to the knockout or high-fat diet mice, show that when you knock out either insulin receptor, both receptors, or feed with a high-fat diet, there's either a trend towards increase of leucine or an absolute increase of the leucine. Interestingly, when we treated with colchicine to inhibit autophagy lysosome system, we see minimal changes in the control in insulin receptor knockout mouse. To our surprise, the double knockout mouse actually showed an increase of the branched-chain amino acid when we're specifically inhibiting one pathway of protein degradation. And there was no significant change in the high-fat diet-treated mice. When looking at the two other branched-chain amino acids, we see a very similar pattern in that they are significantly increased in the McGurko double knockout mice, shown in red, the high-fat diet mice, shown in blue, compared to controls. There are trends in the insulin-only knockout, but again, the pattern of inhibition with colchicine does not prevent the increase of these branched-chain amino acids, other than valine in the high-fat diet-treated control. Next, we went on to look at serum metabolites to see if these changes in the muscle could affect the circulating metabolite pools. And here I'm showing all three branched-chain amino acid content, again, in the same pattern. Just pointing out the control mice compared to the double knockout mice, white versus red, we now see no difference in the circulating plasma amino acids or branched-chain amino acids when you knock out insulin and IGF in the muscle. However, we do see trends or statistically significant increases in the high-fat diet-treated mice, and these actually come down with colchicine treatment. Again, this is treatment of the whole animal, so this will affect other tissues as well. All of these data suggest that if you perturb insulin and IGF signaling specifically in the muscle, you can change branched-chain amino acids in the muscle, but this does not translate to changes outside the muscle or in the circulation. Furthermore, high-fat diet treatment as a model of insulin resistance, you can actually reduce some of the branched-chain amino acids circulating by autophagy inhibition with the drug colchicine. Next, we went on to get a more global approach of what perturbation of insulin and IGF signaling in the muscle will do to the metabolites in muscle using an untargeted metabolite profiling method. We used ultra-high performance liquid chromatography with a quadrupole time-of-flight mass spec to detect these metabolites, again at Mayo. We used unpaired t-test with bootstrapping analysis for a false discovery rate set at p0.05. And annotation identification using Metabolic databases. Here I'm showing the amino acids which were annotated and were either uniquely or commonly increased in the muscle of the three knockout mice that I'm showing. You can see that the double knockouts have a much higher percentage or a much higher number of metabolites which were changed compared to the single knockouts. There's not all that much overlap between these other than the fact that IGF receptor and the double knockout have 4 common. When we look at the decreased metabolite pool, we see actually more overlap between these metabolites. Up to 6 of them were commonly decreased in all 3 genotypes. Although this is a relatively small number of metabolites, we did load all of the metabolites shown here that are changed into pathway analyses database and separated out by the genotype. Here I'm showing the top 5 pathways that are changed in the insulin receptor knockout, IGF receptor knockout, or the double knockout mice. And while none of these reach statistical significance due to the low number of metabolites, I do want to point out a few specific pathways that we did some targeted analyses to prove that these are actually changed. The first I'll start with is the beta-alanine metabolism and histidine metabolism, which was common to the IGF knockout and double knockout. And second, I'll tell you a bit about the glutamate metabolism. So histidine and beta-alanine can be combined into a histidine-containing dipeptide, including carnosine and anserine. These are highly enriched in muscle tissue relative to other tissues in the body and are not found within the plasma because there are specific enzymes which degrade these. When we looked at these content within the muscle, we find that histidine was not changed between the 4 groups, but beta-alanine was specifically decreased in the IGF and the double knockout muscle. Further downstream, carnosine levels were decreased nearly 90% or over 90%, as well as anserine compared to control mice in the double knockout. And these confirm some of the pathway analyses we showed before. Lastly, I'll just touch on a bit of glutamate metabolism in these double knockout mice. Here I'm showing TCA cycle metabolites, which have been shown, which start with citrate on the left and proceed through the TCA cycle to the right. And we can see that there's a relative accumulation of most of the TCA cycle metabolites, with glutamate, which feeds into alpha-ketoglutarate, being one of the highest accumulated within the McGurko muscle. This actually correlated with our studies on mitochondrial respiration, in which we took isolated mitochondria from the double knockout mice and gave them a glutamate malate substrate at the far left. And when we stimulate them maximally with ADP, we see a near 70% decrease compared to the control mice. When you give a complex 2 substrate, such as succinate, both increase, and when you inhibit complex 1 with rotenone, you see that there's no change between the double knockout and control mice. So in summary, what I've hoped to show you is that loss of insulin and IGF signaling in muscle can increase tissue branched-chain amino acids, but not circulating branched-chain amino acids. However, we did observe that high-fat diet-induced insulin resistance increases the tissue and serum branched-chain amino acids, and these can be normalized by autophagy inhibition with colchicine, which likely affects multiple other tissues in the body. Loss of insulin IGF signaling in muscle significantly reduces the histidine-containing dipeptides, carnosine and anserine. And lastly, loss of these two receptors in muscle induces a mitochondrial dysfunction and accumulation of the TCA cycle metabolites. These lead me to my conclusion that loss of muscle insulin and IGF signaling significantly alters amino acids and other metabolites and mitochondrial function, which may confer increased risk of metabolic disease. And with that, I'd really like to thank my mentor and friend, Dr. Ron Kahn, our collaborators on this project, both within the Joslin and at the Mayo Clinic, and of course the Mayo Clinic Metabolomics Resource Corps, which made this work possible. And with that, I'll take questions.
Thank you. Any questions?
Thank you, nice work. I mean, there's good evidence that the adipose tissue is the cause of these changes in plasma branch chain. Is this the plan now to knock out the receptors in the adipocyte?
Yeah, so
is it done already?
It's already done, and actually you get a severe lipodystrophic phenotype. They don't develop adipose tissue very well at all. We haven't looked at branched chain amino acid signaling in this, but it certainly perturbs metabolism very highly when you eliminate these.
Can I come back to the amino acids in muscle? You showed the glutamate effect. Were there any changes in glutamine and alanine? Did you find anything in plasma regarding glutamine and alanine for the enterohepatic, the liver muscle Cori cycle?
Yeah, no, in this sort of like one-time shot, we didn't do any flux analyses. We did not see any differences in any of those circulating amino acids and amino acid metabolites. The glutamine and, you said alanine?
Glutamine and alanine.
Glutamine and alanine within the muscle were not changed in their concentrations. But again, flux analyses may show a very different picture because clearly the protein turnover is very high in these mice.